To begin with, we're going to look at the nuclear membrane. I've drawn a nuclear membrane structure here, the nuclear envelope on the board, this large circle, this spherical structure typically. And in there are a number of little holes. I have these little tiny round circles drawn all over the place. And those are their nuclear pore complexes, And then also, in addition to those nuclear pore complexes on the outside of the nucleus, I'm making these little dark brown spots and little clusters here that represent ribosomes. Ribosomes on the outside of the nuclear membrane. And I won't draw those all day, but we'll put some more in here. So the nucleus has its outer covering here, the membrane we see from the surface, and in that membrane we see these little holes, the nuclear pores established by nuclear pore complexes, and then groups of ribosomes scattered around on the outside, attached to the outside of the membrane of the nucleus. Now I'm going to stop doing this for a second and look at a cross-sectional view of the nucleus here. If we were to cut into it, we would see there's an outer layer and an inner layer, and where we have those nuclear pore complexes, there's a fancy arrangement of proteins that extend from the outside layer of membrane through to the inside layer of membrane there. So that would represent a nuclear pore complex in the nuclear membrane. And then on the outside we have those ribosomes. So ribosomes would occupy the outside surface out here. You'd find your ribosomes out here. So this is a nuclear pore. The structure of proteins here is called the nuclear pore complex. There's the outer layer of membrane. I'll just put outer membrane. And then the inner membrane. There's a space between the outside and the inside. We call that a cisterna. Cisterna. That gap, that space in between the outer membrane and the inner membrane. And then we have our ribosomes. Now, what we can't see from the outside, on the underneath side of that membrane, up against the underneath side of there, we have another layer of material. It consists of intermediate fibers, and it's a little hard to draw in a cross-sectional view like this, but it forms kind of a mesh, a screen of fibers, intermediate fibers on the inside, and that is known as the nuclear lamina. Nuclear lamina. So that wouldn't be seen in this view because it's on the back side of that. So we have a double layer of membrane with a space between called a cistern or cisterna. And we have ribosomes on the outside, and we have these nuclear pores that are established by a group of proteins in a very specific structure. And we call that the nuclear pore complex. On the underneath side of the membrane, the internal side of the membrane, we have the nuclear lamina. Intermediate filaments in a mesh-like arrangement. Now, with all of our organelles we look at, we're going to deal with structure, what it's made up of, and how it's organized, and we'll also talk a little bit about the functions of each of the organelles. So to begin with, the nuclear membrane. We just talked about, introduced its overall shape and structure. It has an outer phospholipid bilayer. and an interphospholipid bilayer, 2 layers, and a space, a cisterna, between. There are ribosomes attached to the outer surface. There are numerous nuclear pore complexes, which are complexes of proteins that form a passageway into and out of the nucleus, a little doorway, essentially, a passageway in and out, the nuclear pore complexes. And there's a nuclear lamina, a mesh of intermediate filaments on the inner surface of the nuclear membrane. So those are the elements of the structure. We've already seen that in the drawing. Functions now, what it does. Containment of the nuclear material. It keeps all the chromatin material together and all the different functioning parts for the nucleus in one place. It regulates movement of materials into and out of the nucleus through those nuclear pore complexes. So we have control of the nuclear contents. There's some function in DNA replication and repair associated with the nuclear lamina, those intermediate filaments, and probably some other proteins on the inside of the nuclear membrane. And the nuclear membrane is also responsible for its deformation and reformation. Remember, when the cell divides, the nuclear membrane disappears, and then when the cell, after the cell division, the nuclear membrane has to reform. So those events are part of the nuclear membrane function as well. So containment, regulation of movement of materials into and out of the cell, or out of the nucleus rather, DNA replication and repair, and then self-deformation and reformation during cell division. That's the nuclear membrane. Our next organelle is rough endoplasmic reticulum, often abbreviated rough ER, rough endoplasmic reticulum. It consists of like plates of chambers with membrane on either side, and on the outer surface lots of ribosomes. So each of these represents a separate little plate of endoplasmic reticulum. These are typically connected. There are membranes layers that connect from one plate of rough endoplasmic reticulum to the next, and to the next, and so on. And it's thought that it often, or usually maybe, the rough endoplasmic reticulum is connected to that outer layer of the nuclear envelope that has all the ribosomes on it. So the outer layer of the phospholipid bilayer of the nuclear envelope would be continuous with these same membranes in the rough endoplasmic reticulum. But anyway, think of these as kind of like potato chip shapes in there, but each of them consists of an upper layer and a lower layer and a space between, another cisternal space between, and on the outside all of these ribosomes we have on here. So if we were to make a section of that and look at it in section, we might see that one of the plates, one of the potato chips runs along there, but it's connected to another one down here below. So you'd have another layer right there, and it might be connected to a third one down below like we have in our drawing up there. But anyway, so you have these different strata or layers of rough endoplasmic reticulum, and the ribosomes are located on the outside of the membrane. This is still outside the membrane where I'm putting them here. It's not to the inside of the endoplasmic reticulum. It's on the outer surface. The little brown dots represent ribosomes. So these are like broad, flat sheets, like I said, kind of potato chip-like. But they're connected. And on the outside we have the ribosomes. So we have membrane on the outside. We have ribosomes outside of that, and then we have the cisterna, that space to the inside. One of the primary things happening here with the endoplasmic reticulum is that we have protein production. We produce protein, so we have amino acids being brought down here to ribosomes And the ribosomes produce proteins that are released into the cistern space of the endoplasmic reticulum. So inside here you have all these strands of protein. Some are already made, some are being made by the ribosomes on the outside. So we're accumulating proteins of different kinds on the inside of the rough endoplasmic reticulum. So the general structure of the rough endoplasmic reticulum, it consists of flattened stacks of interconnected membrane-bound spaces, cisternae, with ribosomes on the external surfaces. So that's the basic structure, composition. Now about function. As ribosomes string together amino acids to make proteins, they are deposited to the inside of the cisternae of the rough endoplasmic reticulum. So we're putting proteins inside of the rough endoplasmic reticulum as they form at the ribosome. Proteins stored in the rough endoplasmic reticulum may become parts of other organelles or cell secretions, so they move on from the rough endoplasmic reticulum to other structures. Next here we're looking at the smooth endoplasmic reticulum. Smooth ER, smooth endoplasmic reticulum. The smooth endoplasmic reticulum, rather than having plates like the rough endoplasmic reticulum, has a bunch of tubular passageways instead of flat passageways that are connected. And they are all highly interconnected in the smooth endoplasmic reticulum. So you have all these tubular passageways that connect and split and so on. And inside, of course, there's going to be a space, a cavity we call the cisterna. So a cistern space on the inside, and of course membrane on the outside. Single layer of membrane surrounding that, and that's about all that consists of it. This network is found out in the cytoplasm of the cell, smooth endoplasmic reticulum. Now let's talk about its functions a little bit. We've already mentioned that the smooth endoplasmic reticulum is a network of connected membrane-bound tubules within the cytoplasm. The functions, we have 3 functions listed here. There could be more, but we've got 3 here. Synthesis of fatty acids and steroids. So we have kind of a storage of those fatty acids and steroids that will be released into the cell or to the cell surface. Steroids mainly that you're thinking of your androgens and testosterone, estrogens, and so on among your steroid hormones. Next, enzymatic role in glucose release and in detoxification in the liver. So it's really a couple of different functions there, but they're both in the liver. And the functions, there's enzymes in the endoplasmic reticulum that are doing these roles. And next, sequestration and release of calcium ions in the muscle. In the muscle, for muscle contraction, you have to release calcium from the endoplasmic reticulum. That causes the whole contractile process of the muscle, and then the endoplasmic reticulum, the smooth endoplasmic reticulum, starts to regather that calcium that was released. So picking it back up again, that sequestration into the smooth endoplasmic reticulum for muscle contraction. So synthesis of fatty acids and steroids, enzymatic role in glucose release and detoxification in the liver, and sequestration and release of calcium ions in muscle. Now for the Golgi apparatus, sometimes called the Golgi body, or just the Golgi. It's a stack of membrane-bound vesicles. Each of these is a flattened sac, one on top of the other. There's one side toward the nucleus down here, and this is referred to as the cis face. Down here on this end, this is the cis face. And this is closer to the nucleus. The top, as we stack these layers of membrane-bound spaces up, as we go to the other side, this is closer to the cell membranes, the outer cell membrane or plasmalemma up here on the surface, and this is referred to as the transface. Each of these is just a cisternal space bound by membrane on the top and bottom. And if you were to make a cross section of one of these structures, you'd find it a little thicker on the margins and thinner through the middle. So they exist in these somewhat like plates that extend through here. Like this. So that's just kind of a cross section through some of the Golgi structure. And materials are added on one side, on the cis face, processed and then released toward the other direction. And some of these form small vesicles as they are released, and some of them become lysosomes, which we'll talk about in a minute. But not much to add to that. So there's always one side of this, I would say it's like a stack of pancakes. One side toward the nucleus, the cis face, and then one side up here closer to the cell membrane, the trans face, and the Golgi apparatus. Simple membranous structure otherwise. Now we've already talked about and looked at the drawing of the Golgi apparatus, but here's a statement of the structure: a pancake stack set of flattened membrane-bound spaces with swollen edges, oriented perpendicular to an axis through the nucleus. The Golgi has a cis face near the nucleus and a trans face toward the cell membrane. So we've been over that already when we talked about the structure. Now the function. It receives proteins from the rough endoplasmic reticulum. So on that cis face where it receives things, that generally comes from the endoplasmic reticulum. Protein products that are carried in vesicles or in some way delivered to the cis face of the Golgi apparatus. In here that we can modify, that means change the structure of the protein material or whatever's in there. It can be sorted so that one region of the Golgi is getting one kind of product, packaged and then released to secretory vesicles, all the way to the, it could be incorporated in the cell membrane to the plasma lemma, or it may form lysosomes with those little vesicles coming off the top of the trans face of the Golgi apparatus. So that's what we know about the Golgi apparatus. It receives, processes, sorts, packages, and then sends out these products, protein products of the cell. Now for lysosomes, as far as a drawing of a lysosome, it really wouldn't look like much. All it is a membrane-bound vesicle. So it would look like a circle, membrane on the outside, a cavity on the inside, and inside it generally contains digestive enzymes. So it's a membrane-bound vesicle containing digestive enzymes. That's what it is, a lysosome. Its function: to bind to endocytic vesicle. An endocytic vesicle is a vesicle that comes from the outside, from the outer membrane, and is moved into the cell. This is usually like a kind of cell feeding kind of thing, or if a white blood cell ingests a bacterium, we would see an endocytic vesicle form, so bringing material into the cell through the membrane, wrapped in a membrane vesicle. So it brings in these endocytic vesicles and breaks down the contents that have been ingested by the cell and release the digestive material within the cytosol. So the cell eats things, essentially, takes them in, and this vesicle that contains what was eaten combines with a lysosome vesicle, and then the digestive enzymes from the lysosome break down the contents of that vesicle, and then the materials are released into the cytosol. Now a couple of other notes here under lysosomes. Autophagy and autolysis. Autophagy there, that refers to eating or breaking down, digesting materials that were maybe no longer useful within the cell. Maybe some of the other organelles that were in there, it can break down some of that material so that those components from that can be recycled within the cell. So that does not result in cell death, that's just recycling material inside the cell. But autolysis is basically the death of the cell. digest the entire cell, and it no longer is an intact living cell in autolysis. So anyway, that's lysosomes. Our next membranous organelle is the mitochondria. There are many mitochondria within a cell. They have this very simple shape. Generally, they can be more rounded or they can be longer, but they're basically like a small sausage shape kind of thing, rounded at the ends and then otherwise cylindrical. There's an outer membrane. We just call it the outer mitochondrial membrane. Then there's a little bit of a space, and then there's an inner mitochondrial membrane. That inner mitochondrial membrane is thrown into deep folds or sometimes simple tubular extensions that extend toward the middle of the mitochondrial structure. These folds that form in here are known as cristae. Now the inner material in here, within the inner membrane in here, this is referred to as mitochondrial matrix. So that's the overall structure of a mitochondria. Now we often refer to this as a powerhouse of the cell because this is where ATP is produced by aerobic cellular respiration. Along these membranes, these inner membranes, we have the mechanism we know as the electron transport chain, an important part of our energy producing pathways, so that's what's going on in here. Also within this matrix we have what's considered it's a form of DNA, mitochondrial DNA. And the mitochondria actually produce their own DNA, replicate their own DNA, and produce more mitochondria all to themselves. So anyway, that's the overall structure of mitochondria. Here's a brief written description of the structure of a mitochondrion. Nearly round to elongated, sausage-shaped organelles with a smooth outer membrane lying very close to an inner layer of membrane which has folds or tubes which extend toward the center. The inward folds or tubes are called or known as cristae. The material to the inside of the inner mitochondrial membrane is referred to as the mitochondrial matrix. So that's what we just described a minute ago. Now look at the function. Here we have three different functions identified, the primary function being the first one here, ATP production by aerobic cellular respiration. That's the main reason why we have them. They can self-replicate, so self-replication is an important function of the mitochondrion, and don't forget they have their own DNA, mitochondrial DNA in there. And that said, they may have a role in apoptosis, which is a programmed cell death. When a cell is supposed to die, when it no longer needs to be there during the developmental process or other kinds of events of growth and change in the body, that's an important part of that. The mitochondria releases enzymes that initiate cell digestion. Our last membranous organelle, the peroxisome, is a very small vesicle, again very much like a lysosome. It's a membrane-bound sphere which contains oxidizing enzymes and catalase. So it doesn't look like much, just a little bubbling of a vesicle inside the cell. Functions, though, it's very vital to oxidation of amino acids and fatty acids, as well as alcohol and other toxic substances. So there are a lot of things that need to be broken down by the enzymes, oxidizing enzymes within the cell. It's an important function of, well, it's necessary for life of the cell. this oxidation process, this breaking down process of amino acids and fatty acids. Oxidation forms peroxides. Peroxides are toxic to your tissues. So in doing these important functions, you're producing something toxic, the peroxides. But the second function down here, breakdown of peroxide. It has what's called catalase, an enzyme that breaks down H2O2 peroxide and other free radicals. So beneficial functions from the peroxisomes, which are just very minuscule organelles within the cell. Tiny little spheres of membrane that contain oxidizing enzymes and catalase.